Okay, so a little bit of my background. So I'm from Chile, actually. I have been working there as a plant pathologist. I have a lot of work that I have done in apple diseases. So then I came here to a Kiev lab. I have been working here for 5 years, and I have been working in different fungal diseases in fruit crops. So here in the lab, we work mainly with grapevines, but we also work with different palm fruits like apples and pears. And also we have been working in other fruit crops like persimmons, and we're working also in strawberries. So, but we mainly focus on fungal diseases in fruit crops. So this presentation, it is about a work that we have been doing about the diversity of fungal pathogens causing apple branch canker and dieback in California. This is investigation that we have been done it for like 3 years in 2021, that we have been collecting samples in different orchards. So here's like a little introduction about our lab in case you don't know it. So this lab is here in Davis at UC Davis in the Department of Plant Pathology. So he's a Kiev, he's the principal investigator here. So what we do, we basically just are like trying to find solutions for different diseases, like how to control them. But first of all, we need to identify them. So in our lab, we receive samples from different people, like we receive samples from PCA, from farm advisors, or for growers directly. And what we do, we make isolation of those symptomatic tissue on a specific media. In that way, sometimes we are able to identify it just based on the morphology of those fungi. Or sometimes we need to go deeper or further just to know what exactly they are. So for that, we just do DNA extraction. We select specific primers, then we do PCR. Sometimes can be like real-time PCR or just regular PCR. And then we need to sequence them. And then finally, we can do a phylogenetic analysis. In that way, we will know that what we are identifying is really the pathogen that we have. So we try to be really accurate when we are doing our identification. So I wanted to start with 2 different concepts that we're going to be talking along this presentation. One of them is canker. So when we are talking about canker, it's basically just localized death or necrotic tissues that we will see like here, for example. And also they're often like circular or oblong, and also discolorated and sunken, like the one that you can see here at the bottom. And also the under the bark, like here's like a pruning wound that you can see here, that after we peel and remove the bark, what you can see here under the bark is all this necrotic tissue. That also sometimes you can see like a well-defined margin that it will be separating like the healthy tissue, like here, and the tissue that is infected. Another concept that we usually use is dieback. So dieback refers to the progress of the disease. So we will refer to a progressive death of the twigs, like in this case, as you can see here, and also a death of the branches. And that generally it starts at the tips, like what you can see here in that picture. And also here, you can see how all this is dead and is start dying from the tip. So those are the 2 concepts that we will be talking. And now I will show you some picture of which were the symptoms that we found in different commercial orchards. So as you can see here in that picture, we saw all this like necrotic tissue, and also the bark start peeling from the branch. And then we peel the bark, and you can see here all this necrotic tissue, that is all the canker that is under the bark, as you can see here. Also here, we saw this like cracking that was all over the branches. Sometimes that cracking can be associated to like sunburn. So that sunburn can cause lesions on the bark and on the wood. So those can be like enter points for the pathogens. So after we peel it, here you can see an elongated canker that is all across the trunk in this case, and also to the other branches. Another symptom that we saw is this particular canker that is really black, and also as you can see here, all the bark start peeling and it start falling off the branch. And here you can see in a transversal cut, it's all the canker all across the branch, and it's really dark too. That is, in this case, we call it like black canker. Another symptom that we saw is here. This is like all around a pruning wound that was made like maybe some years before. And as you can see here, also there is some peeling of the bark, and here all the necrotic tissue. So in this case, it can be two different scenarios that how the infection starts. So it might start by the wound here, or also it may start by the wound that the leaf left after the fall. And here you can see these three cases are like real severe canker that are in the main trunk of the trees. So here we have three different cases with this big canker that are going all across the tree. Okay, so those were all symptoms, but also we saw some signs of the pathogen. So when I'm talking about sign of the pathogen, and when we are able to see the pathogen on the different samples that we took. So in this case here, these small things that are here, those are like fruiting bodies of the pathogen. So that is like one like asexual reproduction structure that they can produce, where inside of them they have all these conidias. So those conidia are those are like source of inoculum that can start an infection in a tree. So here's like a drawing of that pycnidia. So as you can see inside, they have all those conidia. So once they have the right conditions, and in this case, the right condition is that they need water. Like so once they have water from the rain or from the irrigation, what they will do, the pycnidia will hydrate it, and they will release all those conidia and spread the conidia that in case of find like a wound, it can be like a natural opening, or also it can be a wound that was caused by pruning. They can start an infection. So we did a samplings in 2021 to 2023, and we sampled a total of 11 orchards. Those were from 3 different counties, from San Joaquin County, from Santa Cruz, and from Sonoma. And also we sampled like different cultivars, like the one that you can see here in this list. Oh, cannot pass the next one. Oh, there you go. So what we did with those samples, we took those samples from the different orchards and we brought them to the lab. And just to know which are the pathogens that are causing those diseases, we need to make isolation in a specific media that would allow the pathogen to grow in that media. So we put small pieces of the infected tissues on those plates, and then we incubated for 7 days, and then we did morphological observation from those colonies. So here's like an example of the different colonies that we got. So as you can see here, we have different morphologies that are the one that we see. Like here in this picture, different colors, different structure that you can see. So we classify them. So we got a total of 116 isolate, and those isolate, the most prevalent was isolate that belonged to Diplodia. So we got different species of Diplodia, and then it was followed by Euthypalata. There is another pathogen that is really common here in California, causing cankers in different hosts, like in grapevines, in pears, and different cultures. And also some diaportes that also are well known to be pathogens in causing canker. Also Cytospora, and also Phacoramonium and Calmusia varispora. So those are like the main pathogens that we found that were associated to these symptoms. So after we found those pathogens, what we needed to do was to do a DNA extraction just to confirm the identity based on the DNA of this pathogen, and then a phylogenetic analysis. So here you can see the analysis that we did. So from Cytospora, we found Cytospora parasitica. From Diaporti, we found Diaporti camaropsis, and also Diaporti eris. And from Diplodia, we found 3 different species of Diplodia. Those are Diplodia mutila, Diplodia bulgarica, and Diplodia seriata. So from all of them, Diplodia seriata was the most prevalent, and then followed by bulgarica and mutila. However, the Diplodia bulgarica was just found in Sonoma County. It was the only place that we found that species, and also that is like the first report for this species here in the US, actually. After we found all those different pathogens, what we wanted to know is that those pathogens that we were isolating are real, the ones that were causing those symptoms, that disease on the trees. So for that, we need to do some pathogenicity tests just to check if they are able to cause those diseases. So what we did is that with this drill, we did small holes on the branches. We used branches that were like 2 to 3 years old. And after that, in that hole, we insert a toothpick, and that toothpick was covered on mycelium. And then we seal everything with some paraffin tape just to avoid the dehydration and to avoid maybe the infection from a different pathogen. So after 3 months, we went to the field and we cut those branches and we bring them to the lab to measure the lesions and also to do re-isolation from those lesions, just to be sure, just to make sure that the lesion was caused by the pathogen that we were inoculating. So here you can see a picture of the symptoms that we got from those pathogenicity tests. So here the first one is the negative control. And as you see here, we just got like a small discoloration around the wound that we did. However, non-pathogen was isolated from here. And all the other pathogens were able to cause like a necrotic lesion and a discoloration on the branches. As you can see here, all the Diaporti species were able to cause it. Also the Cytospora and the Phacoramonium. Here you see the Diplodias too. And here at the end, Diplodia mutila was specifically this isolate was the most like virulent, so the most aggressive isolate that we got, followed by Seriata that is here and Calmusia varispora that is here. We can see those results here in this graph. And here's like the necrotic lesion, and here you can see the all different pathogens that we use to inoculate the tree. So here we have that Diplodia mutila was the isolate that was the most aggressive, and that was followed by Seriata, Calmusia varispora, and here's another Diplodia seriata. As you can see here, we use more than one isolate for each of the species, because sometimes different isolates that are from the same species can be more or less aggressive. And it's something that you can see here, like Diplodia mutila, the isolate 129 was the most aggressive. However, the isolate 10188, it wasn't that aggressive at that one. So sometimes it's just going to depend on the isolate that we have. So after that, just knowing that the Diplodia species were the most prevalent that were causing canker symptoms here in California, what we did, we wanted to compare just those three species, Diplodia mutila, Diplodia bulgarica, and seriata. And we redo some inoculations just to double check which of them is the most aggressive. So here we got that this isolate also that it was from Diplodia mutila was the most aggressive, followed by these two isolates of bulgarica. And at the very end, we have the two isolates from Diplodia seriata. Another thing that we wanted to do was just to check how they behave based on different temperatures. So we put those isolate at different temperatures, and we measure the mycelium growth on plates. So here you can see on blue we have seriata, in green we have bulgarica. No, sorry, in green we have mutila, and here in red we have Diplodia bulgarica. So here we have that the Diplodia bulgarica, the mycelium growth rate, it's lower than these other two species, and also we can see that the optimal temperature for that species to grow is between 25 and 30 Celsius degrees. And for Diplodia seriata, it is also between 25 and 30, but for Diplodia mutila, it is 25. So we can see that Diplodia mutila and bulgaric will not grow that fast at higher temperatures, like at 30 or also 35 Celsius degrees. It almost will not grow much, and none of them will grow at 40 Celsius degrees. However, 40 Celsius degrees will not be like a little, I mean, it will not kill them, because they can continue growing after you expose them to a lower temperature. So based on those results, we were able to publish this first report of the Diplodia bulgarica causing black canker of apple in California. And actually now we're working in another manuscript where we will include all our survey that we did across those 3 counties. So there we will put all the information that I have been talking today. And also we are preparing another manuscript that it's about some different epidemiological factors that can affect these fungi. So just a small discussion. I just wanted to let you know that these Diplodia species, they have been reported causing canker and dieback, and also they have been reported causing fruit rot during pre-harvest and post-harvest on apple worldwide. So like the different countries that produce apple, they had found Diplodia bulgarica causing canker mainly, and also some of them, they can cause fruit rot. Also, Diplodia seriata was previously reported here in California, causing black rot on apples. And also Diplodia bulgarica has been reported causing canker and fruit rot in not that many countries. So just in Iran, India, Germany, and Turkey. And finally, the species Calmusia varispora that we found causing canker here in California also has been reported causing dry rot, like as this picture here. I mean, dry core rot in apples just in Chile, it has been reported. So our future studies are just we wanted to continue with some epidemiological study just to check like which are the temperatures that will allow the conidial germination, and also if all these pathogens are able to cause fruit infection. And also we want to check different like cultivar susceptibilities. And also maybe just to, I would like to know in case that those pathogens are able to cause fruit infection, like which is like the time when they start infecting fruit. We also have another future study that is about disease control. So that is something that we wanted to do for this winter during the pruning season. We want to try different pruning protectors, like different fungicide, like synthetic fungicide and also some organic fungicide. And that is something that we want to do this winter. And also, yeah, so now we're looking for funding for this research that we want to do, and also finding some locations to do all these studies. Okay, so some conclusions of all these is that all those pathogens that I mentioned before, they were identified here in California causing canker and dieback of apples. Also, the Diplodia species were the most frequently and consistently isolated, and that representative isolates were pathogenic, causing vascular discoloration and necrotic lesions on the apple branches. So that is all. I wanted to thank to our lab, to the Eskalen Lab, to the whole group. I wanted to thank Ellie because she helped us with the sampling. And also I wanted to share to thank to the other former farm advisor from San Joaquin Valley, that is Mohammed Nari. And now, I don't know if you have any question, I will be happy to answer. Thank you.
Thank you so much, Karina. That was fantastic. Really appreciate all of your work on this. I think, let's see, do you need to get going, or do you have maybe 5-10 minutes, or would you like to stick around for my presentation? Because we could do questions at the end.
Yeah, I think that would be better just to do it at the end.
Okay, either way. Okay, cool. So yeah, for the interest of time, I'll go ahead and do my presentation. It's just about 15-20 minutes maybe, and then we'll still have a little bit of time for questions. Thank you. Okay,
sounds good.
Let me go ahead and share my screen. Okay. And we got slideshow. Okay, can everyone see that all right? Okay, awesome, thanks. Okay, so I'm going to present on the management side of things. I'm going to have a lot more photos as well. Karina has some really excellent ones. And so I wanted to include even more because I think the identification side of things is really important. So just to give you a very brief introduction for those who I haven't met yet, I'm the UC Cooperative Extension Specialty Crops Advisor for Sonoma, Marin, and Napa Counties. I work with orchards and veggies, but I don't work with grapes because we have viticulture advisors. And I really focus a lot on soil, water, and pest management, pests including pathogens. And I'm working for the most part in an organic ecological framework because that's the overwhelming approach that my growers are taking, which is very exciting. So I work with commercial growers primarily. And so for those of you who might be home growers, please reach out to our fantastic Sonoma Master Gardeners. Here's their email, their phone number. They work with home growers and are an incredible resource. I've learned a lot from them since I started in January. So please reach out to them as well. So for my outline, I'm going to start by talking about pathogens that cause apple branch canker. I'm just going to skim that though, because Karina went through it so well. And then I'm going to get into the management side of things. So just some brief background. How did this all start? I began this position in January 2023. And in my first week, I received branch samples from an apple grower outside of Sebastopol who kindly brought them into our office. I was new, so I needed help with identification. And so I was encouraged to reach out to Dr. Eskalen, who very graciously said I could send him some samples. So I did, and him and his lab, including Karina, identified the pathogens that she just described. So here's what this looked like, just some more examples of what Diplodia can look like on apple branches. We see cankers, discoloration, some more photos from those original samples. And then let's see. Oh. Okay, so then the lab visited and collected samples to explore all of this further. So here's a keef in the orchard. So we were collecting samples so they could look into this more. And then Karina and Carlos installed the field trial that she described. And then they and others confirmed the presence of many pathogens, including Diplodia bulgarica, and published that first report that she mentioned, which is open access if anyone would like to look at it. So just to look at some more visual symptoms, we see the canker, this reddish discoloration. And in some cases, when it's farther along, you might see these pretty large cankers with these concentric rings. So that's a pretty nice characteristic way of identifying it. So branch canker in general, as Karina mentioned, is caused by a lot of pathogens. I really like this photo because, as you can see with this red arrow, we see this like more reddish orangish discoloration at the bottom, whereas this top area where the bark is sort of similar cut away up top, it doesn't really have that reddish or orange discoloration. So that's a really nice photo demonstrating that. So you might see this reddish brown lesions on small branches just below leaf or twig scars or pruning wounds. The wood tissue underneath is going to be reddish brown, water soaked. And over time, these lesions grow into perennial brown black cankers. It kind of looks sunken like this, and this can lead to that branch dieback. So pathogens survive in this old bark, unfortunately, in the cankers, and produce spores that then can go enter fresh leaf scars when it rains in the fall and throughout the winter. And then over time, like I mentioned, the more extreme case, if you don't really deal with it or prune it out in a timely manner, this can really grow into becoming a pretty large callus. So we have this elliptical, sunken, concentric rings, and that's a more advanced stage of this. So thinking about their life cycle, so these pathogens overwinter on dead branches and their spores spread during rainy, moist conditions. The spores will land on fresh leaf scars, pruning wounds, broken branches, pretty much any exposed plant tissue. They're going to try to colonize if they can access it. And so I wanted to just throw this in here briefly. I know a lot of you probably already know about the fantastic UCIPM website, which has all kinds of helpful information about integrated pest management, or IPM, for key diseases and pests. So for the apple canker, the European canker page for UCIPM, it specifically says in the description for this disease that it is worse in the Sebastopol area of Sonoma County in years with prolonged fall rains, which unfortunately was the case this year. So this is just something to bear in mind. It also mentions that Gravenstein delicious varieties tend to be susceptible, and Rome Beauty as well. So you can always check out the UCIPM page to learn more about this as well, and other pests. So one great question that I got in the registration survey for this event was, how do branch canker symptoms compare with other apple diseases that we might see? So I have this one slide about fire blight briefly. I just want to put this in here to compare symptoms. And as someone mentioned in the chat, that black canker kind of does look reminiscent of branch canker that we've been talking about. However, fire blight is bacteria and it's a completely different pathogen. It spreads much more quickly. So how do we kind of start to distinguish, just using visual symptoms? You might see cankers and you might see some dying branch tissue, but you're also going to see this characteristic hook-shaped wilting or dead shoot tips. So if you see this sort of hook shape at the ends of shoots, this is much more characteristic of fire blight. You also see this blackened center leaf vein as well a lot of the time. So this is, if you see this as well, this is a very good hint that it might be fire blight. Of course, you can also have multiple pathogens on the same tree, but this gives you a nice clue. Two other things to look for fire blight are a trunk canker that has this bacterial ooze. So it has these streaks where bacterial ooze kind of runs down the trunk. So that's pretty characteristic. And then you also, with fire blight, see shriveled blossoms and bacterial ooze in some cases as well. So, you know, if you're wondering, oh man, maybe is it fire blight, you can look for these three additional things that could really help you start to piece together what's going on. You can also, you know, always send in samples to labs as well to find out for sure. Another one people mention is sappy bark, so also known as papery bark. This is what it looks like. Its name is pretty self-explanatory. This is Trametes versicolor. It is a fungi, but it's a minor disease, and it usually only occurs if the tree is already has pretty low health. Maybe it's nutrient deficient, water stressed, maybe there's already another pathogen, more virulent pathogen present. So this one is common, but it doesn't pose as immediate of a threat as these other two diseases. Okay, so I wanted to throw those in there just for comparison when you're IDing things. And now we're going to get into IPM. So that stands for Integrated Pest Management, as I'm sure many of you know. Here's the fantastic UCCE IPM webpage that I mentioned. They have sections for home garden and also agriculture, so that's helpful. So what is a pest? Really broadly, we're just going to define a pest as an unwanted organism that damages plants that we care about. So this could be weeds, vertebrates, invertebrates, nematodes, and, you know, the topic of today is pathogens. So that pathogen could be bacteria, fungus, or virus. If you want to learn more about that, definitely check out the website. So IPM is an ecosystem-based approach to pest management. So it's big picture, holistic approach, and it focuses on long-term prevention first and foremost. And it's really a toolbox approach. So it's the idea that you have this toolbox of strategies that you can use, you know, in combination with each other instead of just relying on one strategy or one tactic. It's responsive and integrated. It's really not prescriptive. So responsive to what you see in front of you, to what you see works and doesn't work at your specific site. So it's context specific. And the goal really is to minimize risks to human health and non-target organisms and the environment. And really, pesticides are only used after monitoring indicates that they're necessary. And of course, always following established guidelines and rules. Okay, so I worked with Akeef to make the management strategies doc that you can find on my website. I put this, actually, I don't know if I did. I think I haven't yet. I'm going to put this website in the chat right now. There we go. So earlier I put my fruit crops website in the chat, and now I put the IPM website in the chat for my particular webpage. So basically, if you go to this webpage and click IPM on the little sub tab on the left, it'll take you here. And if you scroll down, you can find a short article about Apple Branch Canker Management. I'm essentially summarizing this doc and the rest of my presentation. So if you ever want to go back and look at it, it's there. And I can also send this presentation out if you provide your email in the survey that I'll send and share at the end of this presentation. Okay, so IPM for Apple Branch Canker. First and foremost is scouting. So with any pest or pathogen, you want to scout regularly and try to catch it as early as possible. So you can go around and look for dark lesions and cankers on a regular basis. Of course, this is easier when there are no leaves. So now is a great time of year to do it. And then in the early summer, it's a great time for pruning for this pathogen, these pathogens, because the spread of the fungi is much less likely in the early summer because it's not going to be raining as much. And it's still early enough that you could see them before you get to a full canopy. However, also in the fall, you know, you can prune out these cankers in the fall as well. It's kind of less ideal because, you know, as we've already seen, it rains and, you know, that will encourage the spread of disease. But if needed, you could prune out any visible cankers first in the fall and then sanitize between each cut, and then do your annual pruning for tree architecture after that, after you remove all of that diseased tissue out of there. So this is one approach that an advisor up in Humboldt County suggests for this disease. And he said he's found, his growers prefer to kind of separate those things so that you can sanitize between each cut when you're just pruning out that diseased tissue. And then you don't have to think about it when you're doing the architectural pruning after that. So it's one option. But ideally, you'd want to focus your canker pruning in early summer, because that's when conditions are less rainy, less humid, less likely to cause spread. So you're going to remove dead branches, discolored wood, any diseased areas a few inches below the canker tissue, and ensure your pruning cuts are flush against that remaining branch so you don't have a little bit sticking out anymore. You want it to be flush. You're going to want to sanitize pruning tools between each cut, and I know this can be kind of labor intensive, so if you're really short on time, definitely between each tree, but ideally between each pruning cut, because that inoculum, the pathogen, can spread from limb to limb even within the same tree. So ideally sanitize between each cut. You can have a couple different options for how you do that. I recommend 70% isopropyl alcohol, like you see here. This is just what I use when I'm pruning, and I want to make sure I'm not transmitting any diseases. You also have these other options too. Just be aware that bleach can degrade your pruning tools. And then debris removal is a big one. So as soon as you can, practically speaking, it's a great idea to remove all diseased pruning material out of the orchard as soon as you can. That will help reduce the pathogen spread through spore dissemination from those infected branches, like in a pile on the ground, back up to trees nearby. I know Keith really stressed that when we were in the orchard, so that's a big one. Make sure you don't leave piles of infected branches in the orchard. And pruning wound protectants. So Karina brought this up. So you could consider applying pruning wound protectants right after pruning to prevent new infections. And you have a couple options that are organic for protectant fungicides. One is Bordeaux mixture or any other fixed copper material. Here's an example of what that might look like, just as an example. So the way this works for copper is that copper ions denature proteins in the pathogen and kill them. And then you also have biological products such as trichoderma-based products. So trichoderma is a fungi, and it competes for space and nutrients with the pathogens. It promotes the plant's own defensive mechanisms, and it also actually parasitizes the pathogen, which is pretty amazing. So those are two options for protectant fungicides. So zooming in a little bit more on fixed copper fungicides. In general, whenever you're going to apply anything in organic systems, you want to check with your organic certifier first, because different organic certifiers have different rules sometimes. So just give them a call, send them an email, say, I'm thinking about using, this thing for this pest. Is this okay within your rules and guidelines? And once you do that once, you don't have to do it again. So just check first. Of course, you want to read the label really thoroughly, follow all precautions, and apply the appropriate rate for your chosen product. So Bordeaux mixture and these other fixed copper fungicides, they're protectants. They kill fungi and bacteria. So this first application can be made in early fall before the rain. It's a little bit late for that now, but that's okay. If you do have more serious issues though, you can do a second one after most leaves have fallen. So now would be a good time to make a second application, or if you haven't done it yet, a first one's also fine. But now is a good time to do that. Okay, so orchard health, I wanted to just make the point that, using sustainable agronomic practices to promote tree health in general and optimize yield is just going to be good for creating more resilient orchard in general. So it's going to promote strong plant health throughout the season, minimize tree stress, help the trees handle stress a lot better, whether that's from pathogens or drought or anything else. So that means appropriate water and nutrient applications promote plant functioning. For instance, you might want to consider using drip irrigation rather than sprinklers, because Drip irrigation can help increase plant water use efficiency and also reduce that canopy humidity. And so one of the nice things about drip is, you can reduce canopy humidity, which means the likelihood of pathogen spread is going to be a little bit less. But it's also just the most efficient way to water your trees. And I also wanted to say that, not doing any management at all is not necessarily a good thing. So, if you say, I'm just going to let nature do its thing, your apple trees, have been bred for a long time, for many, many years to, rely on some amount of inputs. So this means that you're not going to ensure your trees have enough water or nutrients to really meet the demand of the tree. So, this approach, unfortunately, is great for cankers and other pathogens, pests, invasive species, because your trees are weak and more susceptible to that kind of biotic stress. It can also lead to tree stunting. It's not necessarily great for the environment if you're harboring a lot of pests at your orchard, and your neighbors also might not appreciate having a lot of pests or pathogens that could be at your orchard moving into nearby areas. So it's kind of a regional thing to think about too, in terms of regional effects. So if your orchard is not strategically managed, your trees are going to be unhealthy and weak, highly susceptible to insect pests and pathogens, and not to mention your weeds are going to outcompete your trees for those resources. And one other last quick thing I wanted to mention is that, I see a lot of lichen formation on trees in our region, which makes sense. We have a lot of humidity. However, normally bark, on apple trees tends to shed off pretty frequently, at least enough to prevent lichen from establishing. The lichen itself doesn't actually damage the tree, but it does indicate poor tree health. So your trees really aren't growing at the rate that they should be. So if you're ever wondering, is this bad for my tree? Not really. It just kind of shows your tree's not growing the way that it should be if it were as healthy as it could be. So, I know there's so many things to think about when it comes to actively managing your orchard, whether it's like how you plan your orchard, irrigation, nutrients, protecting it from sunburn with whitewash, pruning, and then, IPM, which is what we've been focusing on. But really the goal is just to do enough to keep your plants healthy while using your resources efficiently. And I just wanted to make the point that doing all this other stuff to keep your plants as healthy as possible, whether it's through water, nutrients, sunburn protection, etc., keeping your plants as healthy as possible will make your IPM job so much easier because your plants will already have their natural defenses. They'll be more resilient. It'll just make your life easier. And it also helps promote regional crop system ecosystem health as well. So that concludes my presentation on the management side of things. I do think we have time for questions now. I'm going to go ahead and let's see, stop sharing. I'm also going to stop recording and we can go ahead and do questions.